Hey guys, welcome back. Today's video is going to be on mitosis or cellular reproduction. We're going to look at different ways that cells divide, but this time we're going to look at mitosis. So let's go ahead and get started. All right, so why do cells divide? So they divide to maintain a workable volume to surface area ratio. So what does that exactly mean? Okay, volume we know is the space inside of the cell, and that would include our cytoplasm. Surface area is the plasma membrane, the exposed environment of the cell. So volume increases faster than surface area. All right, so in cells, we want a larger surface area to volume ratio. Please forgive, that's my dogs in the background. All right. Otherwise, it becomes an issue of getting things in and out of the cell enough so that the cell can work. So let's look at an image. So here we have a large surface area to volume ratio in this smaller block than we have with the larger block. So we want cells prefer the large surface area to volume ratio, that is the smaller piece. So how do we make more cells? So we have to do this by adding cells. Larger organisms don't necessarily need bigger cells, they just need more of them. So we also have to repair and replace cells due to injury or cell death. A healthy cell will divide to replace lost cells, and cells will die for completely natural reasons. All right, and cell division occurs at different rates depending on the organism and the type of cell and where the organism is in their life cycle. Of course, there's greater cell division early on in the human development when when the human is an embryo than they are when they're a lot older. So plant root cells divide more rapidly, and this is an area of active growth. Nerve cells generally do not divide, and this is why nerve damage is an issue. All right, and bacteria divide very rapidly. For example, E. coli divides every 20 minutes, and that makes it an issue when you get E. coli. So how do cells divide? So prokaryotic versus eukaryotic division, what's the difference? In order for the daughter cells to contain enough DNA, DNA must be copied first. All right, prokaryotic division differs because prokaryotes, of course, don't have a nucleus or membrane-bound organelles, so that changes how they divide. Okay, now eukaryotes do have a nucleus, and they have to also have to replicate the nucleus and the genetic material, as well as decide who's going to get how many of each organelle. It's like a divorce. You're going to get a little bit of this, and you're going to get a little bit of this, but it's makes it a little more difficult. All right, so let's look at the life cycle of eukaryotic cells. All right, first you have what's called the interphase. The interphase is the normal part of the cell cycle. The cell spends most of its time in this, and there are 3 stages in interphase. The first stage is G1. G1 is when the cell increases in size and it synthesizes new proteins and organelles. This is really the growing up period of the cell. Then we have the S phase. This is when DNA is replicated, all right? The DNA becomes very stringy and relaxed, and this is called chromatin when it's in this phase, all right? And then at the end of this phase, there are 2 complete copies of the DNA, all right? Each copy of the DNA is called a chromatid, okay? 2 chromatids are held together at the centromere. So look at the image in the corner. All right, we have 2 chromatids, okay, all right, and they're held together by the centromere. All right, and this whole thing together is a double chromosome. All right, and this is what you have at the end of the S phase, because there are 2 copies of the DNA. All right, then we go into the G2. All right, this is when organelles and molecules required for cell division are produced. So we start producing the organelles and everything we need to get ready for division. All right, stages of mitosis. So there are multiple stages of mitosis. Let's look at them. So prophase. All right, prophase. All right, this is when the coiled chromosomes become visible. Those little X's, the chromatids, held together by the centromere. All right, the membrane on the nucleus is broken down, and then spindles or protein fibers that will attach the chromosomes and aid in chromosome movement, the spindles start to form. Okay, the spindles. Then we go into metaphase. All right, metaphase. I like to remember this one because M and middle, okay, metaphase, this is when the spindle of fibers attach to the centromeres and line the chromosomes across the middle. All right, so you can remember metaphase and middle chromosomes are moved to the middle of the cell during metaphase. Then we go to anaphase. All right, anaphase. The spindle fibers shorten, okay, and the chromosomes, the two chromatids, are separated, okay, all right, and we're pulling them out to the sides, all right. The chromatids, of course, are moving to the opposite poles or ends of the cell. Telophase. All right, I like to remember this one by thinking you have to use a telephone to call people who are far away. All right, telophase, the chromosomes get the farthest away and we start to create the two cells. Telophase is creating two. All right, the spindle fibers start to break down at this point, and now we start to see a nuclear membrane form around the clusters. So two new nuclei start to form. They're not completely formed. All right, and then at the end, we have what's called cytokinesis. This is the separation of the cytoplasm and the pulling apart and separation of two cells. Okay, the actual thing where we make two cells is called cytokinesis. All right, animal cells constrict in the middle to pinch apart, forming two different cells. Plant cells also produce something called a cell plate to divide the two cells, and this cell plate is the beginning of the cell wall. And you will have a chance to look at different mitosis animations and activities in class. All right, let's talk about how mitosis is involved in cancer. All right, cell division is controlled by various genes, and the genes that are expressed are different in different cells. So the amount of mitosis, or the amount of cell division that occurs in different cells, varies. Okay, it has to do with contact with other cells and also the available nutrients in the environment. All right, cancer is a disease characterized by uncontrolled cell division. This is when cells start replacing so much, they start to invade other tissues where they're not supposed to be. So how cancer develops, okay, we'll look at that, and there is an extend activity on cancer that is super interesting. But essentially cancer develops because the genes get turned on or turned off, and then we get rapid, uncontrolled cellular division. And those genes could be altered or changed by external factors such as smoking or being out in the sun, or sometimes it just happens naturally within you for reasons unknown. All right, so of course, as we were saying, genes that control cell division, when they don't function properly, we get cancer. And these can be an inherited genetic defect or a spontaneous genetic mutation. And it can also be caused by environmental factors, all right, such as the stuff that's in cigarettes. Okay, look at some of those things. Acetone. Acetone is nail polish remover. Then we have DDT is insecticide that is not allowed anymore. Nicotine, of course, is the addictive part. Naphthalene is mothballs. All right, and so toluene is a solvent. It's often used to paint solvent, all right? And then cyanhydric acid, if you look there, it was used in gas chambers during World War II. So there's some pretty nasty stuff in those cigarettes. All right, so why does reproduction require two types of cell division? All right, so we have asexual reproduction and cell division. All right, asexual reproduction means you have one source of the genetic material, one parent, all right, and the offspring will be genetically identical, and we could also call these clones. All right, there's several types of asexual reproduction. Binary fission, that occurs when unicellular organisms such as bacteria and amoebas divide into two equally sized cells. Okay, so bacteria and amoebas do so by binary fission. Okay, we can also, budding occurs when simple organisms such as yeast produce smaller cells than the parent organism. So let's look at budding. So that is budding. All right, they produce a small thing and eventually it does pinch off. All right, some other types of asexual reproduction, sporulation, okay? This is what happens with mushrooms. It produces spores or teeny packets of DNA for reproduction. All right. And then we have vegetative propagation, and this occurs when a portion of the plant goes through cell division to produce another piece of plant. So if you look at this image of this plant, you can see smaller plants coming off from the extensions, all right, and that's called vegetative propagation. And we have some examples of that with the plants in class, and be sure to ask Ms. Catherine. She can probably show you some great examples of vegetative propagation. Then we have regeneration. This is when parts of an animal, such as an earthworm, undergo cell division to produce a new organism. So if you cut, also we have an example here of a starfish. You can cut a piece off and it will eventually grow into a new starfish. All right, what's the problems with asexual reproduction and what's great about asexual reproduction? One advantage is it's pretty fast and you can create multiple copies pretty quickly. Problems is you have no genetic variation. Everybody's the same as everybody else. And that is a problem because if they're exposed to certain environmental factors, you can't change to respond to those factors. Okay? Mitosis, all right, allows asexual reproduction to occur. So such as when an earthworm is cut in half, all right, mitosis occurs to produce new cells which will reconstruct not the mission portion, the missing portion. All right. I believe this is the last slide, and I will see you in class, and we will be talking more about mitosis.